Hello, my name is David Wang, and I am a PhD student at the University of Pennsylvania. Today I will be talking to you about Chessboard, a model for detecting splicing signals in heterogeneous RNA datasets. Large-scale RNA sequencing projects in recent years have become a tremendous resource for modern translational research, and analyzing such data from large patient cohorts has the potential to provide many novel insights on transcriptomic changes that affect complex disease. However, extracting biologically meaningful signal, which can be defined as a transcriptomic measurement of interest that differs from some baseline level, is challenging due to the inherent heterogeneity of the data. Specifically, as illustrated in this figure on the right, although all patients in your dataset share a common disease state or phenotype, a different subset of genes can be perturbed in different patients, resulting in distinct genetic subtypes of the disease. Therefore, unsupervised stratification of a patient into discrete units characterized by similar transcriptomic perturbations is a crucial step in making sense of the heterogeneity in this data. One domain in which this type of analysis is particularly useful is subtyping cancers based on genetic profiles, such as acute myeloid leukemia or AML, which is an extremely heterogeneous hematopoietic cancer. Now, the traditional paradigm in cancer research has been to analyze genetic mutations as a means to explain disruption of important oncogenic pathways. However, given all the studies to date, this approach has still failed to fully explain the genetic basis of the disease, and thus a transcriptomic-centric view, particularly one focused on alternative splicing where exon inclusion or exclusion can significantly perturb protein function, may provide much more valuable insights, and thus co-regulated splicing events are of prime interest in this study. Now, imagine a data matrix containing some measurement of interest extracted from RNASeq. In the case of splicing data, each row is a splicing event, and the matrix entries are junction spanning reads that are mapped to a splice junction of interest. Splicing is typically quantified as psi, or percent splice in, which is a ratio of exon inclusion, and can be modeled as a posterior beta distribution with priors updated using the junction spanning reads. In heterogeneous data with human patients, there is a high level of individual level variation for each splicing event that must be accounted for. This can typically arise if splicing is misregulated in a subset of patients. If this change occurs in all patients uniformly, the distribution of psi is expected to follow a beta distribution, generally favoring high or low inclusion. However, if we have a scenario where only a subset of patients have misregulated splicing, this results in a mixture of beta distributions, with one component representing a signal of interest indicated by this additional source of heterogeneity. Through this modeling approach, the problem of detecting these splicing events reduces to discovery of a latent tile structure in the matrix, where each tile is characterized by a subset of splicing events and subset of samples, which define an AML subtype. To solve this problem, we proposed a generative probabilistic model for tile finding called Chessboard, which stands for characterizing the heterogeneity of expression and splicing by searching for blocks of outliers and abnormalities in RNA datasets. Chessboard is a Dirichlet process mixture model, which uses non-parametric Bayesian techniques that don't require a priori knowledge of the number of tiles. Now, although similar algorithms exist for related tasks, they all fail to correctly model splicing data. Specifically, many methods model input as real or Gaussian distributed values. Therefore, a psi, which is in the domain of 0 to 1, violate these model assumptions. They also cannot model uncertainty over psi estimates, where we are more confident in psi estimates where we observe many reads. And they also generally cannot handle missing values and integrate them out of the model or require imputation. In cancer data, values can be systematically missing due to mutations. The chessboard algorithm in brief can be explained as follows. It attempts to label each sample or column with a sample group identity, as denoted by indicator C, which follows a CRP distribution, as is the case in all infinite mixture models. Within each sample group, each row is labeled as either 1, indicating a signal, or 0, indicating background, and these labels are regularized using an exponential prior to induce sparsity in the number of tiles. Each observation in the data matrix containing junction spanning read counts is modeled as a binomial distribution with the latent psi value as a parameter, while missing values, which are essentially an indicator for whether the splicing event is quantifiable, is modeled as a Bernoulli, where the parameter, that theta, represents the missingness rate. In this hierarchical model, each row Each tile is characterized by a latent beta random variable for psi and missingness, with priors estimated using an empirical Bayes approach. This design allows Chessboard to handle all the previously mentioned modeling challenges by explicitly modeling psi on the domain of 0 to 1, account for uncertainty in the psi estimates, as well as treating missing values as signals. Chessboard uses a Gibbs sampling scheme to draw posterior samples for the column and row labels. The posterior distribution of the column labels can be decomposed into the posterior beta binomial likelihood and the CRP prior, where the concentration parameter alpha controls the number of tiles. The posterior for the row labels can be decomposed in a similar manner, with beta binomial data likelihood and exponential sparsity prior. To evaluate the performance of our algorithm, we generate synthetic data containing tiles using the generative process of our model as shown in Figure A. We compared Chessboard to two baselines, single-link hierarchical clustering and spectral co-clustering, and evaluate performance using a precision recall metric based on recovery relevance score. Chessboard outperforms the baseline approaches significantly. We then apply Chessboard to beat AML, a real AML dataset consisting of a cohort of 477 patients. We evaluate the performance by showing the tiles found by Chessboard recapitulate known biology, specifically showing that splice factor SRSF1 is differentially expressed between the two discovered tiles, and that there is an enrichment of SRSF1 related splicing events in these tiles. Finally, we assess the translational impact of tiles discovered by Chessboard. Using only splicing events on 70 AML-associated genes, we show that the tiles found in Figure A correlate well with drug response to sorafenib, an AML drug. We observe that gene targets of sorafenib, such as FLT3 and BRAF, are differentially spliced between the tiles, and that the variance explained by mutation status is very small, indicating an AS-associated response to the drug. This may implicate that the role of splicing in AML is more prevalent than previously appreciated, and that one should consider not just mutation burden, but also splicing defects when administering drugs to AML patients. So in summary, we developed a novel algorithm for tile finding in heterogeneous RNA data. Our model outperformed baseline approaches, and we also applied our model to real data and shows that it is able to recover a real biologically meaningful signal. Chessboard is an open source Python tool with an accompanying Python package that can be downloaded from the Biocyphers website, and we also provide an interactive web-based visualization tool called Gambit. So with that, I would like to thank the members of the Biocyphers lab for all of their help and support on this project, and I would be happy to take any questions in the remaining 2 minutes. Thank you.